Hi guys, welcome. In this video, we are going to review and solve IGCSE Biology Paper 6 Alternative to Practical of February-March 2016. Before starting to answer the paper, it is always recommended to read the instructions given on the front page of the paper. So don't forget this during your exams. When you open your paper, keep a rule to read through the whole of the question once. This will give you an idea of what is given to you and what you are supposed to write. You can plan the investigation in your mind as if you are actually performing the same in your laboratory. Also keep a habit of marking the key points in the steps given for the investigations. This will help you to recall the process and interpret the data more easily. You can consider the key points as some variables such as specific volume, specific temperature, specific solution, specific reagent, specific conditions, specific apparatus, specific time period, or anything that you consider to be important. Once you are all set, start answering the paper. Fine. Let's take the first question now. Iodine solution can affect the activity of amylase. The students tested the liquids in test tube W and C using iodine solution without affecting the activity of the amylase. Describe how the students did this. Now, from the question it is evident that iodine is toxic for the enzymes and therefore should not be added to the whole solution in test tube W and C. Otherwise, there will be no activity of enzymes. Hence, students can take a small amount of solution from each test tube W and C in another test tube and test for the presence of starch with iodine, or else student can also put a small drop of water on a white tile and mix it with the drop of iodine solution to test for the presence of starch. Next. The student observed that the liquid from test tube W turned the iodine solution blue-black after 0 minutes, dark brown after 2 minutes, and it remained orange-brown after 4, 6, and 8 minutes. The liquid from test tube C turned the iodine solution blue-black after 0, 2, and 4 minutes, and dark brown after 6 and 8 minutes. Prepare a table to record these observations in the space below. Now guys, remember this part of the paper is actually a cash mark if you follow some simple rules. You are asked to draw an observation table. So consider the 4 rules to draw your observation table. Rule number 1. Check the key points asked in the question to be present in the table. In majority of times, this can be your column headings. Rule number 2. Always write the units, if applicable, in the column headings. Rule number 3. Note down the recording in your table very carefully. They must be given to you in the question itself. And rule number 4. Never write units in the table cells. Alright? So, according to our 4 rules for observation, we can make the table as this and record the readings as like this. Well, this would probably give us the full credits for this question. The next question. Suggest reasons for waiting for 5 minutes at step number 4. Okay, so this is basically to bring the working solution to the required temperature. As the required temperature for the investigation is different than the temperature at which the solutions were prepared and kept. In scientific terminology, this is also referred to as setting up equilibrium. The next, using separate dropping pipettes for test tubes W and C. Now, this is to make sure that there is no contamination as both the test tubes are kept at different testing conditions and mixing the contents would affect the reliability of results. Also, as there are 2 dropping pipettes, the experiment can be done simultaneously, and so the two dropping pipettes are used. The next question. Explain the observations for test tube W. Now, this answer has to be constructed from the observation table. So, from observation table, we can consider that at 0 minute, presence of blue-black color shows starch is present. At 2 minutes, the dark brown color shows that some starch is still present. And after 2 minutes onwards, the presence of orange-brown color shows that no starch is present as the color of iodine solution remains same. So, we can write the answer accordingly. The next question. The students concluded the higher the temperature, the greater the activity of amylase. Do you agree with this conclusion? Give a reason for your answer. Okay, now for this type of question, which are considered as open-ended questions, you can have either point of view. Maybe yes or maybe no. But remember to justify your point. Now, if I consider for the same, then I would probably agree with the statement, as we can see in the observation table that in the warm tube labeled W, the activity of enzyme is faster as the blue-black color persists for a very short period of time compared to tube C. This means that the starch is hydrolyzed more quickly in tube W than tube C. Okay, the next question. There is a source of error in step number 5 of the method. Identify this source of error, and the next one is suggest apparatus that could be used to minimize this source of error. Alright, so what is step number 5? The student added 10 drops of amylase. Okay, now this clearly gives us a hint that adding 10 drops would not have a specific volume because drops may vary in size. So this is our error and it can be easily eliminated or minimized by using syringe or pipette instead of dropper, which dispense a specific known volume of solution only. Okay, let's move ahead. Great. The next question also asks about an error in the method and improvement. State one other source of error in the method used in this investigation. Suggest how to improve the method to minimize this source of error. Alright, now this question is for the general method and not specific to particular step. So therefore we can write any one source of error as experiment was performed only once, or checking the test tubes can cause spillage, or it can be change in color is subjective, which varies from person to person depending on the eyesight, or it can be trying to perform both the sets W and C simultaneously, which could give false negative results. So for any of the set error, we need to give suitable improvement and make sure that it does not contradict to the error. So I would be considering the error as this, and improvement can be this. The next question is, some students stated the activity of amylase is greatest at 40 degrees Celsius. Describe an investigation to test whether this statement is correct. The investigation should be similar to that described in steps 1 to 8. Alright, now this question basically asks you to write an investigation that follows the same procedure given above. So, for this type of question, I always recommend to follow a few basic guidelines. Do remember these guidelines and you can easily write the most suitable answer for this type of question. Number 1. Give the detail of independent variable that you are going to control. Number 2. Give the detail of dependent variable that will change and you are going to record it. Number 3. Give details of some constant variables which you will keep unchanged throughout the experiment. Number 4. A brief detail on methodology to be followed to get the observations and expected results. At this point, be precise as much as possible and do not write too much of the details regarding every step. Be concise. Number 5. Include at least one safety measure. So, if you plan your investigation according to these guidelines, then you can write your answer as this. Guys, these guidelines will always be helpful to write this type of answer in a proper organized way. And I am very much sure that if you follow these guidelines, you can easily write this type of lengthy answers with all the required marking points. Now, the last part of the question 1. Amylase breaks starch down into reducing sugars. Outline how the students could show that reducing sugars are present in a solution. Okay, generally this question can be asked for 2 or 3 credits. Now, you already know that reducing sugars can be estimated with the help of Benedict's test. So, here the key marking points in Benedict's test for reducing sugar includes the use of Benedict's reagent, heating the solution, and observing the color change from blue to red. So, we can write our answer as this. Alright, so this is all about question number 1. Let's start with the question number 2. Leaves exposed to light photosynthesize and produce oxygen gas. A student measured the rate at which the leaves of a pond plant produced bubbles of oxygen gas when exposed to different intensities of light. The apparatus used is shown in the figure 2.1. The investigation was carried out in a dark laboratory. The only light source was the lamp as shown in figure 2.1. The student changed the light intensity by placing the lamp at different distances from the plant. The results are shown in table 2.1. So, here in this investigation, the light intensity is changed using different distances from the plant, and the rate of photosynthesis is measured by counting the number of oxygen bubbles at each distance. The data for the investigation is already given to us, which clearly shows that at lower distance of lamp from the plant, the number of oxygen bubbles produced by plant is higher, while as the distance of lamp increases, the number of oxygen bubbles decreases. Okay, now let's see what they have to ask us for the same. Plot a graph of the data in table 2.1 and draw a line of best fit. So guys, this question is very simple if a little of logical and understanding is applied here. Always remember 4 points for any graph based question. This includes SLAP, in short, slab. Just don't give it to anyone, guys. Okay, so the understanding of slab is S stands for scale or size of the graph. In this point, always consider to utilize at least 70% area of the given graph. The L stands for line, So here always remember to join the plotted points either by using a ruler or connecting each plotted point one by one, using straight line or by making a freehand curve by joining each plotted point. The A stands for axis. In this point, always label the axis carefully and never forget to write the units if it is applicable. Remember the simple rule of labeling the axis, where the independent variable should be on the x-axis and dependent variable is on the y-axis. The independent variable is the one which you control and change during the investigation, while the dependent variable is the one which changes according to the independent variable. Look carefully on the dataset for considering the independent variable and the dependent variable. The last one, P, stands for plot. Be careful in plotting the points. It is always recommended to use sharp pencil for plotting the points with an error margin of plus or minus half small square. You can either use a cross mark or a dot mark for plotting the points. So let's plot our graph now. This is the x-axis, and so we will label it with the independent variable. And this is our y-axis, so we will label it with the dependent variable. Now, if you look back to the data table carefully, the distance of lamp from plant is the variable which is changed by the student for setting up different light intensities, and therefore it is our independent variable. While the number of oxygen bubbles produced by plant depends on the light intensity, and therefore it is our dependent variable. Never forget to write the units along with the labels on the axis. Now the next thing is to scale both the axes so that we can use at least 70% of the given area. Check the highest value in both the table columns and consider scaling both the axes appropriately. Once this is done, plot the graph carefully, checking the values of both variables and marking the point of intersect with a cross or dot. So in the given table, the first set of values includes at 20 cm distance, the number of oxygen bubbles produced are 29. So the point of intersect is this. The next value is at 40 cm distance, 16 bubbles produced, so the point goes here. The third set of values include at 60 cm distance, 8 bubbles produced, so the point would come here. The fourth set of values includes at 80 cm distance, 3 bubbles produced, so the point will be here. And the last set of values include at 100 cm distance, only 1 bubble produced, so the point will be here. Now, after all the points are plotted, the final task is to make a line. We can make a curve or line graph here, but it's already given in the question to make a best fit line. So, to make a best fit line, consider to draw a straight line which usually passes through most of the plots. So, in the given graph, our best fit line would be something like this. Okay, it's all done now. Let's move to the next question. Use your graph to estimate the distance of the lamp from the plant when 6 bubbles of oxygen per minute would be produced. Show on the graph how you obtained your answer. For this answer, look back to the graph and draw a straight line from 6 oxygen bubbles on y-axis to your best fit line. Where this straight line touches the best fit line, draw a connecting line from this point to the x-axis for distance of lamp. The point where the line touches the x-axis is your answer. In the present graph, the answer is 76 cm. Next, describe the relationship between light intensity and the rate at which oxygen is produced by the plant. Okay, this can be easily interpreted from the plotted graph where the general trend shows that as the distance of light source increases, the number of oxygen bubble produced decreases. That is, the rate of oxygen produced decreases as the distance of light source increases. The second point can be any comparative data code with units from the graph. For example, at 20 centimeter distance, 29 oxygen bubbles produced, while at 100 centimeter distance, only one oxygen bubble produced. You can use any one comparative data code from the graph in this point. Next question. Suggest why the student used a water bath in the investigation shown in figure 2.1. Okay, now here most of the students make mistake and say that the water bath is used to maintain the optimum temperature. But actually here the use of water bath is to prevent the heating up of plant from the lamp. So as you can see that water bath will absorb the heat from lamp and will not allow the temperature to rise in the inner tube. The next question. Figure 2.2 shows a photograph of a section through the pond plant as seen under a light microscope. Make a large drawing of figure 2.2 to show the position of the outermost layer of cells and the central core. Do not draw any of the individual cells or air spaces. Okay. Now in this question, you need to draw a diagram called as plant diagram, which only has outlines of different layers visible. Therefore, you need to first observe the given figure very carefully and then plan your diagram. To remember it easily, always keep 4 points in mind stated as SOLD, sold, for plant diagrams. Here, S means this size, which must be larger than the given figure. O means the outlines, which should be clear and single without any overlapping and shading. L means the layers, where all the visible layers should be carefully observed and shown. And finally, D means the dimensions or proportions of the different layers, which must be strictly maintained in your diagram. Now let's see how to draw the plant diagram. First of all, measure all the dimensions of the given figure. Once you have noted all the dimensions, multiply all the figures with a number greater than 1. This will give you the larger size as well as maintain the proportions of your diagram. You can consider any number based on the figure given to you and space provided to you. I generally prefer increasing size by 1.5. So if I increase each dimension by 1.5, the new dimensions I will get are these. Now considering the above figure, I will draw the plant diagram using the new dimensions as this. Remember to use a soft hand drawing. Usually a dotted line for the first rough drawing is applicable. At this point, you can use a compass to draw the circle, but remember in your final draft of drawing, this should not be visible. So the lines have to be as light as possible. Once you have drawn the rough margins, use a sharp dark pencil and draw the outlines as visible in the figure with hand without using compass, annotating the margin lines carefully. Once done, erase out the visible markings drawn with compass by soft hand. If required, redraw the outlines carefully without overlapping. You are done now. Your diagram is larger than the given figure and also maintains the proportions. Okay, the next question. The central core is called the stele. Label the stele on your diagram. So in this diagram, this is the central core. Label it neatly with a clear line. Remember, do not use arrowheads on the lines. And the last question. The diameter of the section in photograph in figure 2.2 is shown by the line AB. Measure the length of AB on figure 2.2. Length of AB on figure 2.2 is as we have already measured, it's 69.1 mm. The actual diameter of the section is 7.5 mm. The magnification of figure 2.2 can be calculated using the following equation. Magnification is equal to length of AB upon actual diameter of the section. Calculate the magnification of figure 2.2 using the information above and your answer for AB. Show your working and give your answer to the nearest whole number. Okay, so the magnification is equal to image size upon the actual size. So the image size in this figure is 69.1 mm and the actual size is 7.5 mm. So upon division we get the answer as 9.21. So converting this value to the nearest whole number will give us the value as 9. So now the magnification is x9 or 9 multiplications. Alright guys, so we are completing the review of this paper. Please give your feedbacks on this video in the comment section below. Thank you very much for watching. Keep practicing and always aim higher.